This morning, we learned about an issue of critical importance to specialty coffee right now. This year, the problem seems to be the specific pathogen outbreak of coffee rust. This organism infects its host through hardly visible spores, making it a divisive and complex foe to coffee. As a science manager, I'm often considering very small details, facts, and organisms, and how these small things influence and cause greater issues in specialty coffee. Well, I'm here today to indoctrinate you with my point of view. This afternoon, we are all going to make a large shift in perspective to the very small. It turns out many problems in coffee are and will continue to be due to limitations in Arabica genetics. We have just seen an example of this with coffee rust. In this afternoon's session, we're going to hear about how Arabica came to be at this genetic disadvantage, what the threats are, what we can do, and what steps have already been taken towards alleviating this problem. Biodiversity has many scales, beginning at the biome and ecosystem level. What we don't usually consider when we view a beautiful, diverse landscape is the microscopic units that are ultimately responsible for that beauty. It turns out there are tiny molecules that code for life, and they have been inherited and evolved for hundreds of thousands of years to stand before you in their current state. What am I talking about? Genes that exist in the DNA code of each and every multicelled living thing. Biodiversity is only possible due to genetic diversity, which is the amount of differences in the genetic makeup of a population or a species. The term genetic variation can also be used to describe these differences. Here you can see two examples of genetic variation within a species, in kidney beans and in a type of marine gastropod. Why does a species need diversity in its genetics? Genetic diversity is needed to ensure a viable and fit population over the long term. The fitness of an organism or population is defined by its ability to reproduce and survive in its given environment. Traits, or the morphological, physical characteristics of an organism, will influence the fitness of individuals. In this way, advantageous traits are passed on to subsequent generations. Genetic diversity is an important mechanism for populations to adapt to changing environments. With more variation, it's more likely that some individual in a population will possess a gene or genes that is particularly suited for a new or stressful environment. Those organisms or individuals will be more likely to survive to reproduce, carrying that trait for many generations. So what do I mean by predetermined? Well, I'm certainly not talking about any divine intervention. I only speak of biology. Plants, turns out, are not sentient beings who have a choice in how they respond to external events or stressors. Plants, in their lives, only have what they are given biologically and where they are stuck physically. In this way, their lives are predetermined by forces outside of their control. The what is what their parents or parent gave them genetically, and the where is where they are rooted down. These two things will ultimately determine the fitness of a plant, and plants can't get up and walk off to better circumstances in their individual lifetimes. Today, we're going to focus on the what. Plants are the way they are because of DNA. DNA codes for all of the biological actions of that plant. And DNA contains genes, which are the individual units that make up the genotype, which is the full genetic constitution of an organism. The genotype is inherited from the parents or parent of that organism. A full set of DNA can be transferred when one parent is propagated as a clone. This is vegetative propagation. In coffee, we often use the technique and term somatic embryogenesis, which is shown here. Or, in the instance of self-fertilization, An individual could have only one parent if it received both male and female genetic contributions from a single plant. So how does DNA create traits? Let's scale down and see. Plants, like all other organisms, are made up of cells. You can see the cells in this cross-section of a green coffee bean, shown here. Multicelled organisms are called eukaryotes, and they have existed on this planet for 500 million years. What you can't see in this dried green coffee bean within these cells are organelles, which are the bodies that help each and every cell exist and sustain life. This image is a microscope view of an onion root tip, and the blue parts are a stain, which shows us the cell's nuclei. The nucleus is the organelle that houses the genetic material. Each nucleus within each cell holds a complete copy of that organism's genome. That genome tells each cell what to do based on its function and location within the organism. Let's take a look into the nucleus. In this slide, you can see that a whole genome fits into the nucleus because first, DNA is coiled around proteins, which are called histones. Then, those coils are wound together to form chromosomes, which you can see in this slide within the nucleus. Plants and animals usually have a pair or 2 copies of chromosomes in their nucleus, and this situation is called diploid. Diploid organisms receive 1 copy of all of their chromosomes from each parent. Humans are diploid organisms because they receive 1 copy of chromosomes from their mother and 1 copy from their father. This image here is a type of microscopic photograph called a karyogram, and it shows a complete set of human chromosomes. You can see clearly 2 copies of each 22 chromosomes, plus a special unpaired 2 chromosomes, which are the sex chromosomes in humans. So humans have a total of 46 chromosomes. However, some organisms have more than 2 copies of each chromosome in a situation called polyploidy. Coffea arabica is a polyploid organism due to its genetic history. In fact, it has a more unusual form of polyploidy called allotetraploidy, because it once received 2 full sets of chromosomes from 2 diploid parents during a hybrid cross between 2 species. In this way, arabica now contributes 2 copies of each chromosomes to its offspring. So arabica has 4 copies of each 11 chromosomes for a total set of 44 chromosomes. To compare that to another plant, onions, for example, have 2 copies of 3 chromosomes. DNA is made up of base pairs, adenine, guanine, cytosine, and thymine, and a sugar and phosphate backbone. Together, the base, sugar, and phosphate make up a unit called a nucleotide. And the order of those nucleotides will eventually make up the blueprint for that organism. Genes are found on this blueprint, and they're found on specific places on specific chromosomes. Those places are called loci. So within the DNA, genes are found at loci on chromosomes. We call different possible versions of the same gene alleles, which means that the genetic code differs at the same place in 2 or more matching chromosomes. DNA is the genetic code, and it codes for traits. But there are a lot of steps between DNA and a living, functioning organism. Inside the nucleus, sections of DNA are read in a sort of mirror image code called RNA, which is very similar to DNA, is formed in a process called DNA transcription. You can see that in number 2 on the slide. Then the RNA exits the nucleus, and another organelle called a ribosome reads the RNA 3 nucleotides at a time. You can see this in number 3 on the slide. The ribosome pieces together strands of amino acids based on that code to form proteins, which you can see in number 4 in the slide. Those proteins then go out to the different places in the cell where they're used for their individual purposes. The proteins, which are built out of these strains of amino acids, form a variety of really important molecules inside cells, including certain hormones and enzymes. This model here on the screen is of an enzyme. Enzymes participate in almost all chemical reactions in cells and assist with forming new molecules. So these proteins are really the building blocks of life. The proteins build the physical and chemical components of an organism. This is how the genotype results in the physical appearance of that organism. The measurable and visual, physical and physiological differences that we see within a species are called phenotypes, and they're made up of traits. You can see two well-known examples of coffee traits in this slide with new leaf color and fruit color shown. These traits are different because there are different alleles present at these genes. Now, let's move on to the other thing that's determined without the sentient consent of the plant, the where. Plants have genetically what they have inherited, but their fitness and survival also depends on where they're rooted. Plants and their genes end up in a particular environment for whatever reason, and some environments are more suited to certain genetic traits than others. So those traits will ultimately influence if the plant thrives or fails in a given environment. Now, it's true that plants have a certain amount of phenotypic plasticity, or wiggle room for different scenarios, built into their genetics. But a plant can only acclimate to a given environment as much as its genetics allow. Acclimation is the adjustment of an organism's physiology, morphology, or behavior to lessen the effect of an environmental stress or some change. This is typically a short-term and reversible process. Acclimation is possible because of phenotypic plasticity, which is the ability of a certain genotype to exhibit variable phenotypes when exposed to different environments. On this slide, you can see two contrasting examples of genotypes, three genotypes on the graph, that either have no phenotypic plasticity or high phenotypic plasticity. You can see that in different environments, the three genotypes express a trait, for example, plant height, shown here, where that plant height either stays static with no phenotypic plasticity or acclimates in the situation with high phenotypic plasticity. You can imagine that the genotypes that have a high phenotypic plasticity would have some advantage when they were growing in a diverse array of environments. However, we also have to remember that phenotypic plasticity has a genetic basis, so that acclimation can only go so far. Let's consider a natural system where species have lived for many generations and are adapted to one environment. Adaptation is a process where a trait gives an organism a certain advantage, and that trait is passed on through subsequent generations, eventually forming a specific genetic constitution that's really suited to one particular environment. For example, the boreal forest on the left. Those boreal plants would not thrive if they were teleported to a very dissimilar environment, such as the tropical cloud forest on the right, and vice versa. This is why there are distinct ecosystem types in different regions of the world. Lucky for coffee, there are many areas in the equatorial regions where it can thrive with human intervention. That intervention is obviously our agricultural practices. For coffee, we are aware that it can grow in a variety of environments, such as sun or shade, shown here. But in all of these environments, we give it lots and lots of help. Now, I'll admit that dispersion of a plant population can only happen in a few ways, and none of them are really predetermined. When we're talking about plants and animals can migrate. On the other hand, plants don't really have an equivalent word to migrate. Plants as a population have what we call a range, and that range basically only changes due to chance events, like dispersion by wind or animal, shown here. But a plant population will only move if a seed lands in an advantageous growing environment. You can imagine that most of these seed transport events are probably short distances, and this is how the range of a plant population can slowly shift over time. However, there have been instances where birds or other animals have moved plants for hundreds or thousands of miles. For instance, if the plant is transported by humans, in this case, a chance event turns into a selection. In agriculture, we make selections based on the trait or fitness of a particular plant that we choose. This is artificial selection or selective breeding. In the coffee industry, we often refer to these coffee selections as varieties, but the more specific term would be cultivar, because cultivars are cultivated variants of a species originating through human influence. You might say, wait, if genetics are predetermined, how do populations change over the long term? Well, I will remind you, evolution does happen in a natural system and over a long time scale. This means that populations can evolve, as opposed to individual organisms, which really only acclimate to a particular environment as much as their individual genetics allow. However, when we're considering agriculture, things are very different. Agriculture is not evolution. In agriculture, we have artificially selected and manipulated traits at accelerated rates. We have brought together plants and species that would have never met in the wild. We have made selections by accident, as well as intentionally, and these have shaped world agriculture as well as the specialty coffee industry. This afternoon, we will hear from a group of talented scientists. From them, we will learn about the history of the Coffea genus and how cultivated Arabica came to be at this genetic disadvantage. Next, we will consider threats to genetic diversity and different ways wild genetics can be preserved. Finally, we will learn about how research is already being conducted to advance the way we think about genes, the environment, and quality, and get the updates from the World Coffee Research Initiatives to alleviate this critical and long-standing problem in specialty coffee. Thank you.